Hello guys, welcome back. Today's gonna look a little weird. We are going to film today's lesson, and you guys are obviously watching that now. We're gonna be talking about genetics. It's just our introduction to genetics. So here we go. We're gonna start off with Gregor Mendel. He is very important. He was an Austrian monk born in 1822. He lived in a monastery and taught high school. If you guys aren't familiar with what a monastery is, it's just kind of like a separate location from civilization with either a bunch of men or women that I don't know, they usually live off the land, and they're generally associated with some type of religion. You'll see a lot of like Catholic monasteries out in Europe. It's pretty cool. Anyways, he taught high school and he experimented with pea plants by crossbreeding plants with different characteristics and studying results. So that was just a bunch of words, right? He's known as a father of genetics because of all of the experiments he conducted that contributed to what we know about genetics today. And genetics entails, all of the different characteristics and traits that we have and what we know about those traits, how we get them from our parents. So basically everything we talked about with crossing over last week and genetic variation, we're going to go into more detail on that this week, and this is where we're going to start. So Mendel came up with a few principles that we're going to talk about. One of those being the law of inheritance. This just states that characteristics or traits are inherited by individual units known as genes. We already know this, we're just adding a fancy term to it. So characteristics, traits are inherited by individual units known as genes. So we get genes from our parents, therefore we inherit them from our parents, thus the title law of inheritance. Vocabulary, gene. Remember that gene is the sequence of DNA that codes for a protein, and then that protein then determines what the trait is. An example, a gene will determine plant height in peas. So that would just be for a plant example. For humans, we could talk about human height or eye color, hair color. There's so many more things that we've talked about. Remember that within genes, we find those individual nucleotides, but then if we expand even farther beyond a gene, we have a lot of genes found within just one chromosome. And remember how many chromosomes all of our cells have. Okay, another principle of Mendel's is law of segregation. This states that during the formation of reproductive cells, so our gametes, our sex cells, pairs of hereditary factors, so genes, for a specific trait separate so that offspring only receive one of those factors from each parent. Okay, one factor from each parent. One factor from each parent. So remember, you'll have a set of chromosomes from mom and a set of chromosomes from dad, 23 from each. And some of those chromosomes are going to code for the same thing. One, mom's going to code for, like, let's say blue eyes, dad will code for green eyes. Remember, that's still the same gene. They code for different information. That's what it means when it says you're getting a pair from each parent. Now we need to bring in a new vocab word, allele. Allele is one of a number of different forms of a gene, okay? So you'll get an allele from mom and dad in relation to the same gene. So that same gene is coding for the eye color, but mom's allele codes for, what did I say, blue eyes, and dad's codes for green eyes. Just making up an example as I go. Now with Mendel, he dealt more with plants. So for him, an example would be more like this flower carries an allele for white and an allele for purple flowers, as you see in the diagram here. Still the same chromosome, okay? They're homologous pair of chromosomes, which means they are the same. They have all the same genes. Those genes carry different information, and the difference in the information we just call an allele. Okay. The next principle we're looking at is the law of independent assortment. This states that chance determines which factor for a particular trait is inherited. So remember last week, some of you guys were asking me questions. Well, how do you pick? It's all by chance. And this is called the law of independent assortment. Remember that we have different ways that our chromosomes can line up when they separate into the two divisions of meiosis. And then we can also have crossing over occur, and essentially all of our gametes are going to look completely different. We just don't know what's going to happen. It's all by chance. Then we've got the law of dominance. This states that when two or more forms of a gene exist, okay, so we have homologous chromosomes, then we have the alleles within the homologous chromosomes from mom and dad. Some forms are dominant, others are recessive. Okay, some are dominant, others are recessive. This is part of that chance as far as how or which allele the child receives is determined. Okay, this is how that occurs. And that's going to be our main focus today, is talking about dominant versus recessive and what that law of dominance means. Well, here we go. Let's talk about dominant and recessive a bit. So some dominant traits, hitchhiker's thumb. I kind of have one. It's where it's where your thumb kind of bends back a bit. It looks like, you know, you've been hitchhiking. And then a normal thumb will just stand up straight. In order to figure it out, you have to push your thumb all the way back as far as you can, though, because if all it can do is go like this, then it's normal. But see, mine can go really far back. Hitchhiker's thumb. Yeah, that's a dominant trait. Some other dominant traits would be free earlobes versus attached. This is one of those ones that I can't really feel, like I kind of have to see it in the mirror to figure it out. I think mine's attached. I don't know. It's not something that you necessarily notice until we start talking about it. Some other would be, a widow's peak is dominant. So a widow's peak is where you have this like little triangle up at the top. You might look in a mirror, see if you have that. It's the image on the left. It's a widow's peak hairline. Some have a bit more pronounced widow's peak than others. Like you can't really tell on mine that much. It's not really like, bleh, but I've got it. It's a dominant trait. And then the straight hairline is actually recessive. I talked about this with one of my classes last week. Polydactyly is dominant. This is where you have a sixth finger on either hand. So, you know, warning, it looks kind of sketch, but you've got an extra finger on either hand. Some people get this surgically removed, but that's not removing the gene. So if this person does end up having kids, because it's dominant, their kids are most likely going to get the gene as well. Some other examples of dominant traits include dimples, which I definitely do not have, tongue rolling. Not going to demonstrate that for you. You guys all know what tongue rolling is. Freckles, curly hair, maybe more than one gene. We've all taught, well, I've told you guys this 1,000,001 times. So my mom has really curly hair. She got it from her dad. So if it's a dominant trait, then why the heck didn't I get it? We're going to talk about that. We are. So we're going to use an example for shapes for pea seeds, and then maybe we'll go back to the curly hair just to demonstrate another example. But generally, when we are writing down the alleles, we try to make it very obvious that one of the alleles is dominant while one of them is recessive. So for shapes of pea seeds, we just picked a random letter, R. It doesn't matter what letter you pick. Okay, it does not. But here we have R. The capital R is what we're going to see as dominant. Okay, so the dominant trait here is round. The recessive trait is wrinkled. Okay, so a wrinkled pea seed. That is a lowercase r. Anytime it's recessive, we see that noted with a lowercase letter. If it is dominant, then it is a capital letter. So we've got, I know some new words here, genotype and phenotype. Genotype is all about the gene. So what are we looking at? Dominant, recessive, is it a mix? Phenotype is what it looks like. So if we're talking about humans, which is just a lot easier, phenotype means face, feature, those sounds, face, feature for phenotype. So that's the characteristic trait, the feature that we're looking at. Genotype is what the gene is coding for. So for this genotype, we have 2 capital R's, so that's dominant. Let me show you what this looks like, because we're going to be talking about this in a box format. We're going to look at this in just a second. But we'll have different combinations to make our genotypes. That's how we're getting 2 R's. So it's like if we have, if the mom, for example, is homozygous, homo meaning same, dominant, homozygous, that would be the same, dominant. So that means we've got 2 dominant traits. So let's say mom has 2 dominant traits, the mom plant, that is, and maybe dad is hetero. Do you guys remember what hetero means? It means different, right? Hetero means different. That's the Latin root word for different. So heterozygous, dominant, means that you have 1 dominant R and 1 recessive R. So they're different. That's where we get the hetero part from. But dominant entails that even though you have a capital R and a lowercase r, that capital R is going to win out every single time. Okay, so let's say dad plant, the dad plant here has a capital R and a lowercase r. So he is heterozygous dominant. That means the trait that he is physically expressing, the phenotype, is still round because our capital R is dominant. Round is dominant. The only time that you will physically see the recessive trait is if you have both lowercase letters. So at that point, you go back to the term homo because it's the same, but then you add recessive at the end because it's a recessive trait that we are physically seeing. So another way to think about this is that the first word here, homozygous or heterozygous, is simply focused on the genotype. So our genotype in the first example is homo, it's the same. Our genotype in the second example is hetero, it's different. And then our genotype in the third example is homo again. The phenotype is all about that second word, so dominant and recessive. Okay, so let's go back to this example of a mom and dad plant. Let's say mom is homozygous dominant. Let's say dad is heterozygous dominant. That's a capital R and a lowercase r. So we have 2 capitals here. We're gonna draw this box to kind of help us see what the offspring could potentially have, the traits that they could potentially have. So if mom plant and dad plant here have children, then they already have 1 opportunity to have a homozygous dominant child. I'm going to use a different color here just to show you how to drag the letters down through the box. So this R is going to continue going down the box, but now we're in line with this R, so that we're going to do our dominant R, then our lowercase r. Can anybody remember what that is? Heterozygous dominant. Awesome. We're going to do the same thing with our other box here on the right. So this R is going to be in this box and in the last box. And then we're going to drag these guys across here as well. So we'll have a capital R and a capital R again, then a capital R and a lowercase r. So let's clean this up a little bit just to see what it looks like. So these are our results. These are the genotypes that our offspring could potentially have. Okay, this is the offspring that our genotypes could potentially have. We've got 2 homozygous dominant and 2 heterozygous dominant. So no matter what, the plant's babies, their offspring, are going to express a dominant phenotype regardless in this example here. Of course, if one of the parents was homozygous recessive, we could be looking at some different outcomes. Now, if we went back to that curly hair example, curly hair is recessive, or excuse me, dominant, right? So my mom's got dominant hair. My dad has straight hair, so there could be some potential my mother's potential genotype. I'm gonna do a capital C and actually let me do a capital H and a lowercase h just because it's a little easier to see. My mom probably has heterozygous dominant, and let me show you why, because of the chances that I could get this recessive trait that is straight hair. And let's say my dad either has to have heterozygous recessive as well, or he needs to be homozygous recessive. So let's say that he's homozygous recessive. And let's just see what the outcome is that we get. We get a capital H and a lowercase h. We get 2 lowercase h's, capital H and a lowercase h, and then 2 lowercase h's. So the chance of me getting straight hair would be 50%. Okay, 50%, because this is the only way I could get straight hair is with the with the homozygous recessive, because it is the recessive trait. But to get the dominant trait that is curly hair, okay, to get the dominant trait that is curly hair, again, 50% chance. And this is probably accurate because I don't think my paternal grandparents have curly hair, so they wouldn't have carried the gene. So he's most likely recessive. But remember, I told you guys that my brother got it, and we have the same parents. So that would make sense to consider this a 50-50 chance of getting my mom's dominant trait that is curly hair. Now, it could be, this could look a little different. We could still get different outcomes, and I could still end up with the recessive trait, but this is just one situation that I, could think of. So we call this a monohybrid cross, and we use this to figure out, what the potential genotypes and then phenotypes for the offspring could potentially look like. And we've already gone through 2 examples, and I just want to reinforce some different ways that we can figure out how to utilize this monohybrid cross, this box, because you're going to be asked in different manners. So for example, we've got this guy right here. Sometimes you may see, what are the potential offspring going to have based on this? And you'll get a capital T by a small t times, and then you'll get the other genotype there. And so what you'll do is what we see drawn here already. You'll just take the first group, and it doesn't matter what side of the box it's on. It doesn't. But we'll take this first group over here, and you'll write one of the letters on the first box and the second letter on the second box. Then you'll take the second group and write it, on top. Again, it doesn't matter if this guy goes here and this guy goes, it doesn't matter. They're going to end up, it's going to look the same anyways. You're going to get the same results. But something else we want to look at is the fact that you're going to see not just the genotype and then understand what the phenotype is after that, but you'll also see it in the form of a ratio. So a ratio is where I was getting those percentages from just a minute ago with the curly hair example. This ratio is, we've got 2 capital T's, so that is homozygous dominant. So that's always going to go first because it's dominant. It's the primary guy. We only see one of him, right? So that's where they got the 1 from. Then you'll put some colons just to separate the letters, separate the numbers. And the next thing we're going to look for is heterozygous dominant. And that we see 1, 2 of. That's where they get the 2 from. Then our last piece is looking for the homozygous recessive. And that is going to be just the 1. Okay, just the 1. In terms of phenotypic ratio, yes, the ratio is going to look different because obviously 3 of these guys still code for the same phenotype. Even though genotype looks different, the phenotype is still going to be the same. So this guy is dominant, this guy is dominant, this guy's dominant. That's why we see 3 right here. Then we have this guy's recessive. That's why we have the 1 down here. As far as how that relates to the percentage, then, you know, it's out of 100 at all times, out of 100. So 2 divided by 4, that'd be half. What's half of 100? 50%. So that's why we get 50% here. And then you can just, you know, 25, 25. I think you guys know simple math like that. Okay, let's take a look at some more examples. Feel free to practice some of these on your own. Now let's take a look at the dihybrid crosses. Okay, the dihybrid crosses. This is when we're getting real fancy. Okay, real fancy. It's gonna look a little more complicated. But it's nothing to stress about. Just try your hand at some of these. We won't really go into too much detail on the dihybrid cross until Thursday, Friday. But this is what it looks like. So you'll have 2 alleles here, the T and the Z. And you can see that we've got a different combination, obviously. We've got T, you know, 2 capital T and Z, and then a capital T, lowercase c. And then, you know, I'm not going to read it because obviously it just gets more confusing. But it's the same pattern. Generally, they'll kind of write it out for you on the box here, this dihybrid cross. And you'll just have to fill in the answers. So if you have T and Z, you'll bring it down the boxes. So every spot here, we see a capital T and a capital Z, capital T, capital Z, capital T, capital Z, capital T, capital Z. Okay, it was just brought down to all of the boxes. Then if we looked at the left, there's another capital T, capital Z. That's where that came from. Capital T, lowercase c, that's where that came from. Lowercase t, capital Z, that's where that came from. And finally, our lowercase t, lowercase c. So everything, again, here is dragged across, here is dragged across, here. It's not that difficult. It just takes some time to figure it out. Now, they generally group the alleles together here, and you may write it differently and then regroup the alleles. It doesn't matter as long as you're able to determine what the correct answer is. But you should definitely give this a try, and then we'll talk more about how this relates to the genotypic and phenotypic ratio again on Thursday, Friday. So good luck. I hope you guys get some good practice out of this, and see you later.
Bye. Bye. Are they gone?